Okay, welcome to this little bit bee chat with Randy Oliver. Uh, Randy's been gracious enough to give us a chat tonight. I want to thank everybody that's coming, and hopefully we all learn something. Randy, if you want to tell us a little bit about yourself, I'd appreciate it, and then we'll go just question and answer. Nothing major.
Okay.
To raise your hand, I think you got to hit participants down there, and it gives another window to the right, and you'll have a raised hand. You got a question, raise hands, and it gives them to me in order, and I'll get to you as quick as we can. You're up.
Okay, I'm a California beekeeper. Been keeping bees since I was a teenager, and got degrees in biology, entomology, and actually was going to go for a doctorate in apiculture, but at the time there were no jobs available. They said you can get in the program, but you won't have a job afterwards. Well, I had a couple of kids at that time, so I built houses for 25 years and built up my bee business until I could take over and just be a beekeeper. And then Varroa mite hit us, and it occurred to me, man, I got a mite kicking my ass, and I got background in biology. Why don't I hit the books here and start learning how to keep bees, from a biological standpoint, and made the mistake of being invited to speak to Oregon beekeepers one year, and everything went crazy. There was this huge demand out there of beekeepers interested in somebody who actually was a beekeeper and understood beekeeping and had a biological background, and changed my whole life. And so I kind of want to just serve the bee industry for the rest of my days and help out beekeepers. It's especially what I find out the internet and social media. There's just so much nonsense out there as far as beekeeping, and a lot of people who give advice based upon ideas or their first year of experience or faith-based beekeeping, as opposed to from evidence-based or from experience-based. So I kind of want to just put out an objective view of beekeeping for the benefit of beekeepers. And I'm now have beekeepers all over the world very much interested in many of the European countries. They say I'm one of the major sources of their information on beekeeping. So that's very gratifying to me. I just got in now, went out. We've got the crew's working right now. I just stopped by out there, but I checked a number of yards for our selective breeding program for mite resistance. I was out mite washing for breeder queens. I'll be doing it when I'm done talking with you. And then we also have a large-scale pollen comparative test of the pollen subs, where I'm testing 7 different commercial pollen subs against each other. And out checking the yards. So we'll tomorrow we'll put the last feeding on before almonds. It's been going for 7 months so far. So we'll see how, when you're feeding your colonies just an artificial diet, which one of the artificial diets actually perform, how they compare to one another as far as colony buildup.
Very interesting. I'm going to start out this thing with a couple of questions, and I get this question a lot, and I've asked it to several different people, and nobody seems to know. Maybe you do, maybe you don't.
Okay.
Here's
one thing you'll see. Different. A good test for anybody, if you want to get advice on beekeeping, if they don't say I don't know from time to time, I would take anything they say with a grain of salt.
That's correct. I agree with that.
A lot of stuff that we still don't know.
Here in Indiana, our winters are funny. I mean, we can go all the way till Christmas before we really get a winter, but we get cold snaps and they go into tight cluster.
Yeah.
And I get asked about feeding dry pollen substitute out. Do or don't. My theory is, I have no scientific proof behind it, is the bees have nothing here from after the goldenrod's gone. They have nothing. So if I give them pollen substitute closer, I figure I'm wearing less of my winter bees out, just going a short distance. Otherwise, they're going to be flying all over the country to get pollen. I know that they could start brooding on it, but they don't like to brood too much, I don't think, on artificial pollen. They got to have some natural in. What's your thought?
Well, it was very attractive, the thought of feeding dry pollen in the fall. And I did measurements in yards of how much pollen, dry pollen, open fed a colony, or the colony on an average would collect in a day, and compared that to putting in the patties. And it was actually cost effective to feed them dry sub out open, because they actually get as much protein into the hive each day as if you took the labor to put it in there. And then I tracked it. I used fluorescent tracer that fluoresced under black light, so I could check the combs afterwards. And they very quickly store it and put it into make it into bee bread, mixing it with any natural pollen that's coming in, and they will ferment it and they will store it. Whereas if you feed pollen sub in a patty form, they don't store one single bit of it. There's no behavior in the bees to move pollen sub fed into the cells for storage. So it sounded like a great idea. And I spoke to a few large commercial beekeepers who had tried it on, well, first I spent a ton of time designing different feeders for dry pollen sub out in the field, and came up with some designs that worked very well. And we were all set up to go into it big time. And then I mentioned it to some large commercial guys who had tried it previously and said, "Oh, Randy, our colonies crashed when we did that. We would never do that again." I said, "Well, that's interesting." So I did a little deeper dive into it. And I noticed the same thing with me, that as far as preparing colonies for the winter, there's a huge hive to hive variation in a yard as far as how much different colonies would take. Some would bring in a lot and some would not. So if you're trying to build your colonies up uniformly, the dry feeding outside does not give uniform buildup. The second thing is, if you monitor the jelly around the young larvae, that tells you whether you have a benefit of giving protein nutrition. So here in California in the dry fall, we look at the jelly, and the nurse bees start cutting back on the amount of jelly they put around the young larvae. So you don't look at the first instar larva, look at the second instar larva. With the first instar larva, the nurses will put a little bit of jelly around the larvae. By the second instar, if they have adequate protein in the hive, they'll completely cover the bottom of the cell with jelly. It's a little harder when they get to third and fourth instar to tell how much jelly there is and how much larva. So the second instar larva you look at, and what we can see is the nurses start cutting back on the amount of jelly around those larvae. And those larvae will still grow, but they're never going to turn into their full potential as a worker because they were on a minimal diet. So what we do is when we test a pollen sub, we monitor how much jelly, if there's a response, and how much jelly the nurses start putting around the larvae. That'll tell you right away whether that pollen sub is a benefit. The best I can tell, the nurse bees won't produce a substandard jelly. If they don't have everything they need to make a fully nutritional jelly, they will just stop making jelly or cut back on the amount of jelly. So the best test, easiest test for any beekeeper to see whether feeding a pollen sub is a benefit is to monitor the jelly in the colony. And within 2 days of feeding dry pollen sub out there, you'll see a response. You'll see them immediately start to put more jelly around the larvae. So if the colonies eat that much, there seems to be a definite benefit for feeding larvae, but that's not what you're talking about. You were talking about them storing it for winter.
Yeah.
So I was curious about that. So I lined up with a Tucson lab and took samples of that stored pollen, stored pollen, pollen sub that the bees had stored in the combs, and combs where they had stored natural pollen. And I shipped them off to the Tucson lab, and they did lab tests and fed bees the two different diets. And they found out there may be actually adverse effects on the bees on the fermented pollen sub, where they take the dry sub and store it in the combs, as opposed to the natural pollen that is fermented for the same amount of time. There's only been one study, only with my combs. We published it in a scientific journal. That study definitely needs to be replicated, but it certainly supported the the field observations of several commercial beekeepers that they can actually may be able to harm the colony by feeding large amounts of dry sub in the fall and having them store it as fermented bee bread. How's that for a simple answer?
That's the answer I've been looking for, because people ask me, and I usually keep pollen sub out, and I've had some trouble over the last few winters, and that may be my culprit.
That may be it, and it's a certain. I have a list of unanswered questions that I will guarantee you have never been answered scientifically or by field trials. That to me is just stupid. That as much as big as the bee industry is, and there are many scientists out there looking for something to research to publish, that they don't do it. And one of the problems is, if you're a researcher, especially a young researcher, the young ones don't have much experience with bees in the first place. You can see it in the photographs. They got these brand new white bee suits on. They got the veils and the gloves and all that, and brand new white beehives. They don't know the beginning about beekeeping. That's not their training. So it helps a lot for these researchers to have a mentor who actually knows something about beekeeping. The second thing, for a career advancement, they need to get published, and they need to get published in higher tier journals. Higher tier journals are very picky about what they want to publish. They want to publish stuff that's sexy, that's cutting edge, that will get media attention. Doing a feeding trial on how dry-fed pollen sub affects your colonies is a big question for a beekeeper making his living, but it's not going to get you published in a high-tier journal. So there's very little scientific interest in doing that. Unfortunately, the USDA and the universities, the land-grant universities, which for any other animal husbandry or plant husbandry, do a lot of this kind of applied research that is not sexy, but that benefits the industry. We have very little of that in this country in beekeeping, and it's an absolute shame. Our industry is so far behind any other livestock or agricultural industry. It's a joke.
It is. Yeah, that's the best answer I've got, because everybody else has basically said I didn't know, which I didn't know either, and that would give me an idea of what's been going on. Kind of interested in your mite-resistant bees. What kind of studies or raising you doing on that? You got a certain breed, or you just testing bees?
No, what I did is I realized we're not that the final solution to Varroa is to have mite-resistant bees. If we had followed the track of the beekeepers in South Africa when Varroa arrived, and not used any synthetic miticides or mite control measures whatsoever, most of us would have gone out of business, but we would have had resistant bees within a decade. In South Africa, it took about 6 years. So right now, South Africa, when Varroa arrived, it infested all their colonies. They did lose a lot of colonies. But if you look at the meetings of the South African beekeepers now, or you look at their annual reports or their monthly journals, they don't even mention Varroa. They don't control Varroa. It's in all their colonies, but it's not a problem for them because they have resistant bees by just simply not doing anything. They let nature take its course. Most other countries, or all of the countries, did not do that. We jumped right on, and in the integrated pest management pyramid, where you only use a synthetic insecticide as a last resort, we just jumped right to the top of the pyramid and used that right off the bat. Well, that stopped. That could be used for a stopgap measure, but it makes it much harder to breed for stock that is resistant. And what's happened is the large commercial breeders who are producing most of the queens used by beekeepers throughout the United States, they're not scientists. They don't want to have a really complicated program. They don't want to have to go out and dissect thousands of larvae or do a whole bunch of freeze kill tests or do all this kind of work. So they're resistant. And the second thing is they sell every queen they can produce right now without any claims of mite resistance. So there's no incentive for them to do that. So what I do is I try, and I know them all, and I talked to them and I said, here's the deal. The first one of us that has truly proven mite-resistant queens, you are going to be scrambling to catch up, because that's what the demand is going to be for. People are going to want those mite-resistant queens that are proven tested mite-resistant queens. So here's what I'm going to do. I'm going to forget marker-assisted selection. I'm going to forget instrumental insemination. I'm going to forget closed population, all that. I'm going to try walking the walk and see if you use old school breeding, selective breeding, that most every livestock breed we have and most plant cultivars were done by old school breeding, where the breeders didn't know anything about genetics, they didn't know about DNA, they didn't know about inheritance. They simply selected for the traits that they wanted, and after enough generations, you get those stocks. I said, I'm going to do that, and I'm going to use only one simple test, and that is the mite wash. And the thing that changed everything is to build battery-powered portable mite washers, so you can just knock out mite washes fast. And what I did then is I said, okay, here's what I'm going to do, guys. I'm going to walk the walk, the good, bad, the ugly. I'm not a salesman. I'm not going to try to do anything. Let me see how much it would cost and what kind of success I will have. And I found out to put 1,000 hives into a selective breeding program cost about 100 man hours of work a year. It's very, very small amount and very simple. I mean, very little record keeping. And the only thing you need to do is to do the mite washes. And with a mechanical agitator, that takes the strain out of those. So a few years ago, when somebody asked me, Randy, are you thinking that people should mite wash every single hive in the operation? And I said, no, that's crazy. No way in the world would that ever happen, that the commercial guy's going to mite wash every hive. The last 3 years, my sons who are running the operation now say, okay, let's do all the mite washes, and we mite wash all 1,500 hives in our operation in June. And they want to do it. And they want to do it because it saves them money. There's more benefit to doing it than there is cost. The small cost in doing it pays for itself in one, mite treatments that you don't have to put in, so you save the cost on the mite treatments. And number two, by identifying the mite bomb colonies, that 10% that's going to just spread mites throughout the rest of the operation, you've identified them, you take care of those, and then mite management is much easier throughout the whole rest of the operation. So once a year, we do indeed mite wash every single colony in our operation, and we find it to be cost effective to do that. Then the only thing we need to do for the selective breeding program past that point is any of the colonies that are below a threshold. So for our late June wash, anybody who's got a mite count of more than one mite in the alcohol wash is out of the program for a potential selective breeder. They get treated if necessary. But the ones that have counts of 0 or only 1, they get a paper tag put on top of the hive, and we come back a month later and we wash those. Now that's a much smaller number. That's been around 250 or so. And then a month later, and then at that point, if their mite counts are going up, they get treated, the tag gets ripped off, and they just get run right with the rest of the bees. So we're not moving colonies around, we're just doing that in the yards. And then we just do that throughout the year. And by the next spring, those that are still, they're all considered potential breeders up until next spring. And if they've maintained very low mite counts for the entire year, and they're a gentle colony and productive and look good, then they are a breeder. So we started out our first year with less than 2% of our colonies graded as potential as breeders. It went up to about 5% the next year, and the year after that, we were up to about 7%. Last year, we were running about 10%. We had about this time of the year, late fall, early winter, we had about 200 out of our 1,500 we started with that still needed no mite treatment at all. We finally narrowed it down to 70 of the colonies for breeders that we actually bred off of. This year, I'm tallying up the numbers now. It's a little bit less this year for some reason. I'm not quite sure why. I don't think we're going to make the 10% again, but we're nowhere near the 95% being that are resistant, that I would think about claiming that our bees are mite resistant. The frustrating thing is heritability. So we have colonies that are absolutely mite proof. They can sit in a yard with colonies collapsing of the mites around them, and they will maintain a mite count of 0 or 1 mites for the course of the entire year, with mite drifts and everything coming in. They are big, productive colonies, often the best colony in the yard, gentle as pussycats, and they're perfect. Their dreams come true. But you raise daughters off of them, and only a few of the daughters colonies exhibit the trait. So I'm not really sure just what's happening, what kind of magic combinations of alleles of different genes they have to get, whether there's some transgenerational epigenetic inheritance, which is passed on via the jelly, whether some of that resistance came from a drone that the queen mated with, and so that 1 or 2 patrilines of daughters exhibited those traits. And in that case, the queen would not pass those genes on, other than to, if she made it with 1 drone out of 20, then 1 daughter of hers out of 20 would actually carry that allele. So I'm trying to figure that out. But I've seen proof of concept. I can assure you, there are colonies, you can have colonies that are absolutely resistant to varroa. I just found one, I checked 3 years. It's been out in my yard, in my experimental yard, where we bring in the highest mite colonies for doing experiments. So they're just flooded with mites all year. 3 years without treatment, and colony still going. So I can guarantee you, it's possible to have gentle, productive, totally mite-resistant colonies. The trick is trying to figure out how to breed for them so that you can increase that to 95% success rate in the operation.
Now, when you raise daughters off that, the ones that have mites, is that the ones you put them queens in requeen with? Is that how you're trying to do it? Get rid of them?
We requeen our entire operation every spring. We split everything we have when we come back from almonds, split them into nukes. Half of our income is from selling nukes each spring. So we make about 3,000, 3,500 nukes every spring, sell about half of them, and start our operation with the other half.
Okay, so you got all new queens from that year's breeders.
Absolutely. And they made out with drones from last year's queens. So the genetics of your queens from the previous year supply the genetics for the drones in the next year.
But by I got a question. By requeening all of them, even your perfect queens that you had, and by requeening them, maybe you're losing the herd genetic side. I mean, they're getting it from the drone side, but maybe the drones are not transposing it on out in generations. Maybe it's coming from the queens.
Well, the drone genes all come from the queens. So like I said, your drones, those genetics that come in from the queens from the last year. So we're shifting now to doing what's called progeny testing, where you keep track of all the daughter colonies of a particular queen, follow them, and then go back to the original queen, which means you keep your queen from the previous year, and you see what percentage of daughters. I just finished with those washes just this month, and we were disappointed. We had a couple of yards where what you look for is whether half the daughter colonies exhibit resistance, and we had 2 yards where it looked like that was the case. But in our last alcohol wash, most of those got eliminated from the program, so we did not have a high amount. So I'm in the middle of this. I'm going to stick with it and see where we're going. It's just, here's the thing: when you see these incredible mite-resistant colonies, completely mite-resistant colonies, you want it to happen. You want to just make it, you want to keep going with the rest of them. We just don't know how long it's going to take.
You'll get there. We got a question here from John Curtis. We're gonna, John, you have to unmute yourself, and you're up.
Hey, Joe, you got one in the chat too.
Okay, this is just a follow-up on the dry pollen sub. Since you don't feed it in the fall, is there any good time to feed that throughout the year?
No. I mean, if they eat it right away, it wouldn't be fermented, and this is research that's just crying, and then this to be done. And I'll tell you right now, anybody with a number of colonies could do this research, or any club could do it. Here's the trick: most beekeepers don't know how to design a project, a scientific experiment. So if you want to ever do an experiment, make sure you find an experienced scientist who can help you with the experimental design and write down a protocol for you to follow. So when you're done with it, you haven't just wasted your time. They actually can learn something from it. So that's research that needs to be done. I would, from what I've seen is those colonies that do gather that dry pollen stub, they immediately start producing more jelly. So if they're not storing it, I would assume that it would be a benefit. But again, there haven't been enough experimentation to know.
Okay.
Thank you.
You want to read that question from the chat, Jason? I don't have it up.
Sure. It's from Kenny in West Virginia, or actually he may be in Ohio. I'm not sure. Kenny Bach. He says he's been fortunate enough to attend meetings in Ohio and West Virginia where you were a presenter. My question is about the oxalic glycerin shop tiles. Any result or future observations?
Oh yeah, I've got 4 articles in a row coming out in the American Bee Journal, and then once they're published, I can put them up on my website. We did extensive testing of them. We made thousands of them this year. Instead of the shop towels, we use Swedish cellulose sponges, and I have a dozen new matrices, absorbent matrices, to try out this next spring to see what the ideal one is. And we got very, very good results with them. They outperformed the other treatments that I compared them to, which was HopGuard and Formic Pro, which are the only other 2 that are approved to use while honey supers are on. And what we found out is it doesn't take a lot of oxalic acid if you apply it early in the season and it's on the colonies. Just a very small amount, that trickle effect, does the job very well. The other thing is that it's a long-term effect, and we really don't fully understand what's happening, and it may have something to do with changing the mites' behavior. Some of the data I have indicate that it may prevent the mites from re-entering cells to reproduce after they've come out. They just stay on the adult bees and they don't go back and reproduce. So it takes a month and a half to 2 months before you really start seeing the full effect. By 70 days, we're getting incredible effect. The other thing is, because it's a longer term treatment, if there's much mite drift coming in from outside, from other hives, it can overwhelm the efficacy. So last year, we ran trials in our yards around our home. And then we also had the opportunity to have 4 different yards that were totally isolated with no other honeybees within flight range. 3 of them were up in the high Sierra Mountains at around 6,500 feet, where there's no population of honeybees that survive up there. And the other one was in an isolated little valley up in the middle of the desert, in irrigated valley in the middle of the desert in Nevada, where we were the only honeybee colonies there. So there was no outside mite drift coming in. And the oxalic sponges performed spectacularly there. They just took the mite counts down to 0. It was just absolutely amazing. So the possibility of this treatment, and what I'm seeing now, is you could put it in early in the season before you put your honey supers on. And when you pull your honey supers off, you've got, it doesn't get into the honey, and you got no mites in your hives unless they came in from immigration from outside. And so they can handle a certain amount, but the less immigration from outside, the better. There's some research showing that it also will work well over the winter. So we've got some colonies now that we've been trying it over the winter to see whether there's any adverse effects. So that's what I'm looking for, is not just benefit for controlling the mice, but to see if there's any adverse effects. During the summer, depending upon the ratio of oxalic acid to glycerin you use and the amount of substrate that you put in the hive, we don't see adverse effects. There's 2 main ratios used. The Argentinians have a formula where they dissolve the 1 gram of oxalic acid in 2 milliliters of glycerin. And the advantage of that one is that the oxalic acid stays in solution. So when you put it on a piece of cardboard or something, it stays wet and sticky, okay, from the glycerin. You don't see any crystals of oxalic acid at all. What we find is that those are really messy to handle. You get that acid solution over your hands and all of your equipment. And if you put too much in the hive, the high amount of glycerin transfers the oxalic acid onto the bees too quickly. And you can see all your bees starting to buzz inside the hive with agitation. They may beard up out in front of the hive, and you can see some brood kills. So you have to be careful not to put too much surface area of that into the hive. The formula that we have been using and that we like better is the ratio of 1 gram of oxalic acid to 1 gram of glycerin, instead of a milliliter to a gram. So 1-to-1 weight to weight solution. To make that solution, you do have to heat it to dissolve the oxalic acid. You have to absorb it into the matrix while it's still heated. And then overnight, that oxalic acid will crystallize out because it's a supersaturated solution. By the next day, you have a towel or a sponge that's slightly rigid with white little crystals all over it, but it's not nearly as messy on your hands or equipment, much easier to handle. And it doesn't bother the bees. And what it does is then, as that glycerin absorbs moisture from the higher humidity in the cluster, it sucks in water. That water then dissolves some of the oxalic acid, and there's a slow release of oxalic acid over a very long period of time. So in the sponges, we were using sponges that contain 25 grams of oxalic acid, but maybe only 5 grams of that was ever distributed out into the bees over the course of the 70 days.
Okay, so how big a sponge are you putting that on?
It's
surface-wise?
about 3 inches by 8 inches. Or 3.5 by 8. The sponges that we used came 7 inches by 8 inches, and we cut them in half, so you get 3.5 by 8 inch. And putting one of those in did not give enough surface area, did not give that good a mite control. Putting 2 of them in gave spectacular mite control. We were concerned because the 2 of them contained a total of 50 grams of oxalic acid, a fair amount. But the reality was, when at the end of 70 days, when we scraped them out, 40 of that grams of oxalic acid was never released from the sponge. This was still in the sponge at the end of the trial. So it was only a smaller amount. So now I'm going to try some thinner matrices that maybe hold the glycerin long enough, but perhaps the bees will chew them up at the end of the trial or something like that, or be easier to remove.
Okay, it's 3.5 by 7 by how thick?
They're about 1/8 inch thick.
The ones you're using, 1/8 inch?
Yeah. Well, that's the ones that I used. I'm testing 12 different substrates for my tests to compare those two. So we like those better than the shop towels, but they're still not perfect. We're still looking to see what else could be used.
Okay, we got a bunch of questions now. Got everybody's attention now. Let me see here. Jim Duffy, you're up. Unmute your mic.
Hey, Randy. Stay.
Yeah.
I've been watching your videos and they're awesome. Now, putting shop towels in a beehive, you recommend putting them through in between the kind of in the central part of the
First thing,
cluster.
I cannot recommend anything because it's illegal.
Okay. All right.
So don't try to trip me up here, okay?
Okay, I gotcha. All right. I want to use the shop towel on a nuke.
Uh-huh.
Any way of doing that?
What we did is, for experimentally, we made strips with the sponges and hung them over the top bars like you would a hop guard strip, and that worked well. When I tried that with shop towels, a lot of the hives, they would chew through the shop towel right at the top edge of the top bar, and the strip would fall down to the bottom board and would not be effective. With the sponges, we didn't see them doing that. So if you used a matrix that they did not chew through, that works well. And that's what the IUN cap does. They use a strip of cardboard that is hung over the tops of the frames. The reason that we don't do that is the first year that I tested it with the cardboard strips, it was really amazing. We got good results. And we were in a big bee yard, and I said to my son, Eric, who just walked in a second ago, I said, Eric, look at this. We figured out this new mite control method. It's great with these strips. And he goes, well, how many strips in a hive, Dad? I said, well, 4 didn't do it. It took 8 strips per double deep to get good mite control. And he goes, let's see, 8 strips times 1,500 hives. And he was really quick at math. He goes, that's like 2,400 strips that we're going to make and insert one at a time and then pull out one at a time. He said, never going to happen, Dad. Figure out some other way. So simply because we're commercial beekeepers and we're not going to put 20,000 strips into our hives, I looked for a way of that we just crack the hive and just lay something across the top bars of the lower box. I started with shop towels simply because commercial beekeepers have long experience of putting treatments into hives using shop towels. So it'd be an easy sell for beekeepers. And my hope was that the bees would then chew the shop towel out afterwards and disappear it. What turns out is when you add glycerin to the shop towels, most colonies don't chew them out completely. And you wind up with a mess with a bunch of propolis over it. You have to scrape off with a hive tool. So we shifted to the cellulose sponges, which the bees don't tend to propolis into any great extent. And they're much easier to remove. They just put a thin band of propolis around the outer edge, but they soften up enough by the end of treatment that they don't come out in one piece sometimes. So now I'm got some matrices with synthetic fibers in them to strengthen them up. So I'm looking for something that will be easy for the beekeeper to pull out of the hive once the treatment is over.
Is that a look to be almost as good as Apivar or hard to tell?
Well, Apivar is, there's a lot of resistant mites out there to Amitraz. Amitraz has been used so strongly by the commercial beekeepers for so many years, and they're the ones that supply you the queens, and that's been the only treatment they have used for years. So they've been in a selective breeding program for not only good queens, but for, you tell me.
My resistant Amitraz.
You got it.
Okay, Kevin Eaton, you're up. Unmute yourself and ask your question.
That's Karen. Hi,
Oh, I'm
Randy.
sorry.
That's okay.
Hi, Karen.
Amongst all you guys. I'm from New Hampshire. Randy, I have a question I haven't been able to find in my searches about royal jelly or jelly in general. So the 3 days of royal jelly to all larvae, the 3 days after, I see everywhere brood food or whatever. But isn't that still jelly? Or I see reference to pollen and nectar. So physically, is bee bread and honey or nectar put into larva cells, or is it eaten and turned into a different formula of jelly?
It is eaten and formed into jelly. The research that I've seen and my own research where I've used fluorescent tracers is that any pollen that ends up being fed to the larvae was inadvertent. It was an inadvertent contamination on the mandibles of the nurse bees when they were eating the pollen or the bee bread that just happens to fall off when they're secreting the jelly. The jelly is the food, and I don't even use the term royal jelly.
I know you always use jelly. So
jelly. The nurses make
jelly. Drones get jelly. Larva gets jelly. But
right.
Is there a distinction?
Now, of that jelly is made from 2 components, one from the hypopharyngeal glands and one component from the mandibular glands. Both of them have to come out as a thin liquid or the bees would not be able to get it out of their glands through the tubes. So they take those 2 thin liquids out, secrete them between their mandibles. When they come together, they react like epoxy does, and they turn into a jelly. So the jelly is formed from 2 components added together in their mandibles. They can adjust the proportion of those 2 components slightly, plus they then disgorge from their crops nectar or honey from their crops. So the queens, they put a whole lot more sugar into it. And I just downloaded just 2 days ago all the data from a Chinese researcher who analyzed the jelly very carefully over time for the workers and the queens. And that's on my to-do list maybe this next week to graph out how those different proportions change in there. But it's essentially jelly though. They're going to shift it a little bit. And with the queens, they feed a lot more sugar, and that allows them to be fed better and much more. If you've ever raised queens, you see the workers load up those queen cells with far more jelly than the larva can possibly eat in its early instars. And after the cell is capped over the queen cell, then the larvae continue to eat and gain weight at the very end after it's capped over. So, and that jelly is not fresh jelly. That was jelly that was put down the first 3 days when they were being fed exactly the same components as the worker larvae.
Okay, so just to be crystal clear, because I'd really love to see a scientific paper on it, just so that I can keep that in my repertoire. Bee bread and honey or honey is not directly fed to larvae. So when people say they change their diet to bee bread, they're not physically taking bee bread. Thank you, Ray.
Correct.
Thank you so much.
Yeah, and we tested that out with feeding with pollen sub in the patties and putting in a fluorescent tracer and then building a blackout tent, raised our forks up and put black plastic layers in there, made a blackout tent. One of us could sit inside there with a black light, and then we could pass the frames that we pulled out of these colonies after feeding fluorescently labeled bee bread into there, and we can inspect to see where any of that fed pollen sub wound up, and not a trace of it in the combs. Occasionally you'd find a little flake inside some stored honey because it fell off a worker's mandible, but there was absolutely none ever given to the larvae.
Thank you.
Okay, that's the books are not always right, are they?
The books are, there's a number of glaring errors in almost all the textbooks because somebody said something because it made sense many years ago, and because it makes so much sense, everybody else just cites it. For example, all the books say that Nosema apis causes dysentery, and then they'll cite another scientific paper. But if you go back to that scientific paper, that paper just cites that somebody said that. There's never been one single study ever published on the planet Earth that I can find that has ever demonstrated that Nosema has ever caused dysentery. But all the books say that. But in fact, I had a journal editor from a European journal asked me if I would review a paper on Nosema and dysentery, and I said, okay, I'll do that. And I said, but I'm not, because she called me Dr. Oliver. I said, I'm not a PhD. And she goes, that's okay. We've been recommended. You should review this paper. So I reviewed the paper, and it was all about how you could identify whether the bees had Nosema ceranae or Nosema apis by looking at their poop, by looking at the dysentery in the hive. And there were citations saying, oh yeah, that they cause dysentery. And I said to the editor, I said, look at the citations. Nowhere in those citations, nowhere, anywhere in this paper have the authors ever cited that there's a study that showed that Nosema caused dysentery. And even in their own study, they had like 50 colonies or 100 colonies, all of which had dysentery, but Nosema was present in only about a third of them. But they wanted to say, that Nosema caused dysentery, even though the 2/3 that also had dysentery had zero Nosema.
Yeah. Okay, Todd Prather, you're up. Unmute yourself.
All right. Hey, Randy, how are you?
I'm fine.
We've chit-chatted a couple times via email about strips and stuff. I do have another question though about mites, and going to the opposite side of the equation. Down here in Fresno County, in the last couple of years, they have been spraying or releasing sterile mosquitoes into the neighborhoods instead of flying a plane over. Has there been any research about maybe making a sterile male mite, or is it even possible?
It wouldn't do you any good because the mites mate with their brothers inside the cap cell. So you could release all the male mites you wanted, but they'd never make it inside a cap cell.
Okay.
Oh, I was actually involved with research with a startup company producing sterile mosquitoes, and I was taken down to Brazil and taken to Italy, working with this startup, and we were able to develop RNAi that we targeted the genes involved in spermatogenesis of the male mosquitoes. We were able to put that into their larval food and produce male mosquitoes that were sterile from what they ate as larvae. And that and we did a demonstration project in Brazil and showed that by releasing these, we could control Zika virus. So very interesting project. A little aside I was involved in.
Is there any possibility on the mite side to make it impossible to produce offspring?
Yeah, the bees in Norway figured out how to do that. And so this is what I'm saying about if you do a breeding program for mite resistance, most of the people who have done these tell the bees how to do the job. They say, we're going to select for this specific trait, whether you're doing varroa sensitive hygiene or whether you're doing biting. On the other hand, if you just let the nature take its course with random mutations and just tons of generations, they come up with ways that you would never in your wildest dreams would have thought of. So let's look at a little bit molecular warfare here between the bees and the mites. The mite is such a perfectly adapted parasite. So let me go back to you right now. Let's just say you got all the protein in your diet from eating salmon, and your cells turn over constantly, so you're rebuilding your whole body all the time. If you ate nothing but salmon for protein, would you turn into a salmon?
Probably not.
Probably not. No, probably. You would not. We know that because we know people that eat nothing but salmon, okay? And the reason is when you eat salmon protein, your body digests that salmon protein, breaks it down into amino acids. And then your body reassembles the amino acids into human proteins. Varroa mite doesn't do that. They have evolved so specifically for living on the honeybee that they take honeybee proteins directly and use them without digesting them. In order to make eggshells, they need proteins called egdysteroids. And there's 7 egdysteroids that they need. They use the honeybee egdysteroids, and Varroa has given up evolutionarily its ability to make some of these egdysteroids. They're entirely dependent upon the honeybees to produce these egdysteroids. The bees in Norway have downregulated their production of some of the egdysteroids, and that prevents the mites from laying eggs.
So obviously it's incumbent on the bee to figure that out.
And who would have ever thought? Who would have ever thought to breed for that? So this is what's so wonderful, incredible about biology and evolution. It's going to do things that you never in your wildest dreams could have imagined. So in answer to your question, is there a way to affect varroa reproduction? Well, nature figured out a way for those bees to do that.
Okay.
And with feeding and using with the oxalic and glycerin, that appears possibly also be doing that to keep the mites here. And here's the point I make continually. In order to completely control varroa, you don't have to kill a single mite. You don't have to kill one single mite to completely control varroa. All you need, because they're all going to die of old age. All you got to do is keep them from reproducing before they die. Okay, you don't have to kill them. You just have to keep them reproducing, and that's what the oxalic acid and glycerin may be doing. It will kill some, but it appears that maybe it keeps the rest reproducing.
Is there anybody in the United States studying that deal to keep them from making the shell or stuff for the egg shells?
Not that I know of.
Yeah, it looked like that would be something somebody ought to be jumping on.
Oh yeah. First thing, you need a fair number of colonies, and it'll probably take a long time, and it's unlikely you're going to get a sexy scientific paper out of it.
Yeah, but if you had to fix varroa, you would be.
But that doesn't increase your job opportunities as a scientist.
Yeah.
Okay?
There again,
We're-
there's a little bit of problem with academics then, right?
No, it's a problem with us beekeepers thinking that we have to have somebody else solve our problems. If we want to come up with the money and pay them to do it, they would do it. It's our problem. The beekeepers we're the dumb ones by not getting the money where it should go. And I had a big run-in with Project Apis M about that. I got kicked off the board as scientific advisor because I felt that we should be funding projects that were directly applicable to beekeepers. And instead of the scientists telling us what we should be doing, the beekeepers telling the scientists what they should be doing. That did not fly well with some of the researchers.
While we're talking with money, you want to tell them how they can donate to your projects?
Oh yeah, you could happy to take donations. You can just go to my website, Scientific Beekeeping, and click the donate page. I just got called by USDA this morning. I was out in the bee yard saying, okay, so can you confirm that you're going to donate $5,000 to this bee lab for the project to do this analysis of the gut microbacteria? And I said, yep. You just tell me where to send the check. So I'm $5,000. I'm going to be kicking out for doing that. I spent 2 days ago, I spent over $500 shipping costs with the dry ice for samples going in. So yeah, we have some pretty big scale expenses. In the pollen sub trial we're doing right now, we had 144 hives in that, 18 in each group, 8 groups of 18. So we knew in the sham treatment that just got sugar patties alone, that they would never make it to almond pollination. They're not going to grow. We knew that in advance. Okay, we get $225 a hive. That's 18 hives we are not going to get paid for. So that's lost income in order to run this trial. We still had to maintain those hives, do everything else, but we're not going to get any return, and we're not going to get any nuke sales. So we're not going to get that $300 worth of nukes or that 225. So that's a $500 loss per hive. So that's expenses that it costs. So these experiments cost money. And what I use is just the donations from beekeepers to offset my out-of-pocket costs.
Very good.
And so everybody who's donated, I appreciate it.
Sharon Walter, you're up. Unmute yourself. There you go.
I was trying to put a 3-year-old. Let me get a 3-year-old out of the way, and then we'll take it. Okay. All right, he's on his way out the door. When you talk about the viruses that are causing a higher impact to the bees' mortality, I think that over the last couple of years we're seeing overwintering. Now, just to give you some background, we're in upstate New York, very cold environment. So I think in our environment, they either live or they die, and it happens very quickly. We can't limp anything along. If they don't go in real strong, we're going to lose colonies. So we're in the last couple of years, we're seeing higher rates of mortality with some of those smaller colonies, which we didn't used to have any trouble getting through. Mm-hmm. So now, we're wondering whether, EFB, is becoming more and more prevalent, and we're having more issues with it. I know this year alone, I've opened some of my smaller hives and been concerned, like, obviously we always have that, the elephant in the room with, is this AFB or is this EFB or what are we looking at? And I feel like we're seeing more and more EFB in our hive.
Across the world, this is a common thing. In California, it hit us really hard several years ago. And a lot of the commercial guys were really complaining about their hives not building this stuff. And I said, guys, take a look in there. You've got EFB. You just haven't ever noticed it. And I said, you're not necessarily going to even see it because a lot of us have fairly hygienic bees now. So you're not going to see any symptomatic larvae in the hives unless you look at several combs. So like when I do field sessions and for trainings, I'll open a hive, I'll take one look at a comb and say, oh, this colony's got EFB. And they said, how can you tell? And I go, here, let me find a larva. Well, not on that comb, not on that comb. I may have to go through 3 or 4 combs before I finally find a symptomatic larva to show them it does have EFB. But those colonies that don't grow in the spring, that's the most common thing I see. So we had it really bad here in California, and now it's pretty much disappeared. We see the occasional case. So it can just go in waves like that. Same with chalk brood. It can go in waves where you see it in just every single hive. And then the bees, through this, either genetically or transgenetically, what we're finding is that the bees via the jelly can pass on immunity to their offspring, so that this is the way possibly they deal with these evolving viruses and fungi and bacteria. What we saw during when there was the first shortage of bees back with CCD, and there was a shortage of bees for almond pollination, and the USDA allowed us to import package of bees from Australia. I had been over looking at hives in Australia the year before, and I said, oh, look at this. Your chalk brood looks slightly different than our chalk brood in California. I can see it's chalk brood, but the color is just a little bit different in the cells. And we thought that was interesting. Well, the Australian bees can handle that chalk brood by themselves. You bring them to California and you expose them to California chalk brood, and every colony just crashes from chalk brood. So the little bit different in the strain variation between the chalk broods was enough to difference between colonies being able to handle it and not being able to handle it. That's what's happening like with COVID right now, where we're finding out, oh, well, the virus is mutated. Yeah, that's what these pathogens do, especially the RNA viruses mutate very quickly. And once they mutate, they can get around the immune system. And that may be what you're seeing. And what we're seeing is the first thing is with the viruses, when you're doing PCR, unless you have the primer, you can't even identify a virus is there. So a number of years ago, Dr. Michelle Flanagan, she's a virologist, University of Montana, said, hey, Randy, do me a favor. Could you pick out a handful of colonies that before almonds that are just doing very poorly and not building up, and then an equal handful of colonies that are doing very well side by side, and send me samples over time from those two sets of colonies. And what she found out, there was a virus called Lake Sinai virus in there, and that was the difference between those colonies. No one had ever found Lake Sinai virus in honeybees before then, or had any idea that's a problem. Since then, just in the last few years, we've realized Lake Sinai virus is often one of the main players for bee mortality. So, but it had been there all along, but unless you have the primer to look for it, you don't know it's there. So we're in a learning curve of all these viruses. Our deformed wing virus, A strain, is largely being replaced now by the B strain, which used to be called Varroa destructor virus. So that may be something that's happening in your area. So I'm not surprised on this. The main thing that you can do is to minimize most of the viruses in the colony, is to make sure you have very, very low mite counts, virial counts from middle of August on. So the last rounds of brood that the bees rear in the fall are not infected by viruses. That's your best bet for getting into the winter safely.
So that's, we had very, very low mite counts. So when you talk about a primer for what's causing it, we're seeing kind of crazy weather patterns. So we're, in the past, maybe we've had a more consistent weather pattern where we're typically going into very, very cold climates. And, for us, it could be October. And then we have snow on the ground from sometime in October, right through into April. And then we go in, we've got end of April starts to pop and we go. Yeah. Now, this past year, I mean, we had a 25-year, 30-year drought, last year. So, I mean, that's obviously going to take a huge toll on the bees. Absolutely. Bringing back resources and having some significant issues with it. So I'm just wondering, you talk about a primer for some of these things that are underlying, whether they've always been there and it's more of an environmental cause for some of these that are happening.
Oh, I'll tell you right now, in California here, we normally have extra bees that we don't contract for almonds, and we hold on to them, and then we get premium price at the last minute when the growers are desperate. This year, we are struggling just to fill our normal contracts. And what happened is we couldn't figure out why our colonies, we couldn't build them up in the fall like we normally do. And then I downloaded all the data from some personal weather stations around those apiaries and compared this year's data to last year's data. What I found out is we had unusually hot, often 5 to 15 degrees Fahrenheit higher weather in September and October than we had the previous year or the historical norm. That's the time when we're normally building up our colonies to go into the winter. And we're right when you start to get above that 95-degree temperature, you're stressing Apis mellifera, Ligustra, and Carnica, those strains, the Italian and the Carniolan bees. They are not adapted for those temperatures, and they just shut down. So you can't build them up. The second thing is if you have a drought and you don't have the pollen flow coming in you're expected, it makes a huge difference. So yes, environment, it'd be a huge thing. But you got to realize humans and honeybees have had 10,000 years of a very stable environment on the planet Earth. We humans have decided to change that environment suddenly and get used to it. The environment is going to be changing a lot. You're going to see weather patterns that you have never seen. Here where I live, where we used to take the boys tobogganing here on my property on the snow every winter. We haven't even seen snow here for a few years. So it's just in my lifetime, the weather here has changed and the climate here has changed tremendously. The plant blooms have changed, the plant timings have changed. We're actually taking bee yards that we've had for 35 years and we're giving them up and moving to higher elevations because the yards that used to be good at that elevation are no longer good for the bees.
Yeah, well, we typically have queens coming back from mating flights sometime in the first week of May, really early in low numbers, but we can typically get them coming back then. We had 6 inches of snow on the ground the 16th of May. So that pushed everything out for us. So the bees were struggling at that point, even just to have enough resources to survive, then to come back to our summer, which was a complete drought. I think it was just kind of a perfect storm of like just terrible weather.
Absolutely. What we're seeing now with climate change is these oscillations of the jet stream that instead of moving across quickly, they now stall. So now where there'd been a minor break in the rain or a minor bit of cold weather sits there for a month. And that's what we're seeing. That's what we're going to have to get used to is these slower oscillations where you're going to see long-term dryness or long-term wetness or long-term coldness. These are all parts of climate change due to all this carbon dioxide we're pumping into the atmosphere.
Good. Well, I don't want to cannibalize this call, but I do have, one last question that kind of goes along in the same lines with this. We've heard in New York State that there's been an increase in the number of AFB outbreaks that we're seeing.
Yeah.
So again, obviously, from what we see and what we hear is that the bees have a certain threshold that that they can tolerate, and then when that spore level gets too high, then it becomes a problem. Mm-hmm. So again, maybe because of this year with the weather impact, could be another reason why we're seeing AFB, which normally wouldn't have been an outbreak, had been tolerated in the past. Are you guys seeing more and more? I mean, I know it's kind of the elephant in the room. There's a lot of people out on YouTube that have thousands and thousands of hives, but you never hear one of them talking about AFB or any of these viruses because, I kind of give them they'd feel like it'd be a black eye, when in reality, it's beekeeping. And I think it would be better if more people were open about it. We do our best. We do our best to try to, when we're selling our nukes, to try to talk about the things that could come down the line and what to see, and please call us so that we can get them in touch with the right people. I think sometimes when people are putting videos on YouTube and putting things out there, they're trying to put their best foot forward, but it doesn't necessarily do the best thing for the beekeepers and the newbies.
So what we see is here in California, in the Bay Area, San Francisco Bay Area, there's a huge population of new beekeepers, many of them wanting to be treatment free. And those beekeepers who actually want to make a living with bees, it's almost impossible because of the outbreaks of American foul brood down there and the huge drift of varroa mites down there. So it's a very much of concern. I grew up with American foul brood, and it was a huge problem. We came up with a zero tolerance policy if we see one celibate in a hive, that all those combs get burned and the equipment gets sterilized. Years ago, I selectively bred for bees that were resistant to American foul brood using the freeze-killed brood test. And I had colonies that you could drop a full super of solid foul brood infected brood on, and a few days later, you wouldn't be able to find a single cell. They were just so hygienic, they would just clean it right up and they'd be fine until you had a pollen dearth or some stress. And you see it pop back up in those colonies. So all these, so there's pathogens and there's disease. The colonies always have the pathogens. They only have disease when the immune system cannot keep the pathogen in check. So yeah, you can have American foul brood spores in there all year long, but when the colonies get stressed, then it can form disease. The problem with the commercial guys is many of the commercial guys have been using tylosin antibiotic off-label. Tylosin is only allowed to be used in a beehive and dusted in sugar, never fed in syrup. And the reason is because it doesn't degrade in the syrup and it winds up contaminating the honey. There's lots of loads of honey now that are rejected by the packers because of tylosin residues from this off-label application by the commercial beekeepers. They're doing this every fall, which means that they're just suppressing foul brood in their operation. So then they swap all the combs around. Pretty soon you have homogenized foul brood combs throughout all 20,000 of your hives, and you're stuck. If you don't put the antibiotic on every single year, your operation comes down with foul brood. If you sell nukes, when those buyers, unsuspecting buyers, buy those nukes, and if they're not treating with antibiotic, then they can flare up. I was out at one of the universities a couple of summers ago, and they had bought 50 nukes from some commercial beekeepers. And when I opened them up, there were brands from 3 different outfits on the top bars of these frames. They were just a hodgepodge of junk frames. And when we looked through a few of them, I had a video camera on my forehead. I finally said, I got to turn this off. We've seen every disease known to beekeepers in these nukes that you guys just bought. We've seen American foul brood, we've seen European foul brood, we're seeing a chalk brood. They were just absolutely ugly. Well, these are nukes that were sold to a university.
Wow.
Yeah.
I got a couple more questions here. You good for it, Randy?
Yeah.
Okay. Mark's iPhone, you're up. Mark, you there? Okay, we got Susan. I can't pronounce your last name.
It's Churchill.
Okay, go ahead.
I just was curious if you've done any studies on the heated mat things that they claim will kill the varroa mites.
Oh, funny you should mention that. I'm writing the article. I've been working on it all morning.
Oh, good.
I tested some myself, and the results that I got were not very good. After the treatment, I went into those hives and dissected out the drone brood and dissected out the worker brood and counted the number of live mites and counted the number of dead mites. And it was surprisingly poor kill of those mites. So then I did a deep dive into the previous research, and then I put out the word to beekeepers who have tried this, and I'm analyzing, been spending all week now analyzing data sent in from other researchers. If any of you guys have done that, I've got another week before I have to submit the article. If anybody's got data, the interesting thing is you ask the manufacturers for supportive data that shows the efficacy, and it's not out there. There's just, it's really hard to find that. So I'm analyzing anybody who's treated and has actually taken either mite drop counts or sticky board counts or track those hives over time. And it's very little information I've got, but I'm summarizing it all in the article, and I'll be sending it off for publication the 20th of this month.
Well, thank you, because I tried researching it also to see if I could find any studies that had been done, and I had not been able to find any. So that's why I was curious. And the other thing about that was that I had anecdotally heard that it killed the brood. And so I didn't know if that was the case too.
I did not see it kill an appreciable amount of the brood. Here's the deal, is it's a very, very narrow temperature range that's critical. It's somewhere around that 106 to 108 degrees. And you're right at the edge. 108 will start to that will kill it will kill the drone brood, and it can hurt some of the worker brood, and also maybe hurt the bees. 106 doesn't hurt the bees much, but it doesn't kill very many of the mites. The second thing is when you use the treatment, most of the adult bees go out and hang out outside the hive in the beard, and they're not hot, so it does not kill the mites that are on their bodies. And roughly during the summer, roughly half the mites at a time are on the adult bees and not in the brood. So it does not deal with those. The thermal treatment that works well, they have in Germany, you take your combs of brood, brush all the bees off of them, and put the combs of brood into this chamber for 2 hours. It takes it up to the high temperature. It does kill the mites, and then you put the brood back into the hive. But then you still got all the mites on the adult bees.
Okay, Mark looked like he's back on his iPhone. Mark, go ahead. He's not there. Mr. Jason Braggs, I'm sure you've got a question. You're the scientific mind in our little bee buddy. I'm sure you've got something you want to ask Randy.
There's a lot of things. Too much to eat up the chat with, but I did have one thought back when you were talking about jelly, in the queen cells. A year or two ago, I decided I was going to try to artificially test if I could artificially raise queen cells in an incubator.
Mm-hmm.
So I started out the larva, and this I designed a fake or false cell cap, 3D printed it, had it ready. It just clips right over a JZBZ cup. Loaded them with royal jelly, and started them, grafted them, started them in the incubator, and they grew to a fairly decent size for about 5 days, and then they all died off on me. And so I was trying to figure out and the only thing that I'm coming to the conclusion was that the nurse bees touch those so many times before they're capped. They're probably cleaning feces and a lot of other stuff out of there.
Wouldn't be feces, because the larvae don't defecate.
Okay. So what are your thoughts on that?
Don't know. When they artificially rear workers, they don't use straight jelly. They use jelly with a little bit of yeast extract in it. And I don't know that's odd why you would have to do that. But that is something that is done. And I haven't heard an explanation of why that is.
Well, that was a curiosity of mine, but that's just something I was playing with, and I was curious as to why they would get to a certain age and then quit. Or just die.
I mean, one possibility also. Did they turn color when they died?
Yeah.
Did they turn black when they died?
They did not turn black.
That sounds like
no.
They're not turn black. Okay, that's not okay. They turn black. That's typically black queen cell virus, which will often kill off queen cells. So I couldn't tell you.
Yeah, no, they just turned a kind of a yellow off-white color.
Okay.
But I was prepared, when they got large enough to spin their cocoon, I was just going to slip that 3D printed cap over, see what happens. But never quite got that. I may go back and revisit that someday.
Yeah, I haven't heard of anybody else even trying that. That's interesting.
It's just the thought. But that's all. I don't want to eat up your all's chat.
Mark says he's back there. He got his mic unmuted now. Mark, you're up.
Okay.
Yeah, sorry about that. I've got a question about the high humidity. I live in a very high humidity area, and I saw where you was you there?
Yeah.
Okay. And you was talking about with the sponges or the strips and stuff, that a high humidity area was a problem because of I don't remember what part of the process caused that, but I
didn't say it was a problem. I said that was a suspicion. When a paper was just published out of Veracruz, Mexico, where they use oxalic acid, they use aluin cap formulation on hives at extremely high humidity, and they worked just fine. I also am publishing a study out of Virginia by a high school student who got a permit, so I could collaborate with him. and he got 88% efficacy in short term in Virginia at high humidity. So I think the humidity thing was a red herring.
Oh, okay. And the other question I have is, are you still using like thymol and stuff like your sugar waters and stuff for preventing fermentation and stuff? And that's
For
it.
preventing fermentation, we use bleach in our sugar. If the weather's hot, when the weather's cool, we don't bother. During the summer, if we're feeding sugar syrup, we put a little bit of bleach in there, and that keeps the mold from growing. We still use thymol for mite control. We still, despite the oxalic acid being so promising, I still like rotating treatments to hit the mites from different directions.
How much bleach you adding? What's your ratio on your bleach? You got any idea?
Yeah, we put about a cup in a 55-gallon drum. So that's about a half teaspoon per gallon.
Okay, I do too. I just wanted to see where I was standing at. Jamie Kelsey, go ahead. Jamie, you there? Unmute yourself.
Sorry about that, Joe.
Mm-hmm.
This goes back to what Jason was talking about with that capped, trying that capped cell. Could it be that the reason that they dropped off like that might have to do with the sugar content in that royal jelly? Didn't you say earlier,
that
Mr. Randy,
could
that
well be, yeah.
Sugar?
And well, Jason, where did you get the jelly from?
Harvested out of queen cells that I raised.
What age queen cells?
They were probably a day before capping or so.
Okay, and when you harvest the jelly, take the top layer, or do you take the bottom layer also? Because they're different. The top layer is different from the bottom layer.
Okay, I took it all. I just harvested the whole cell.
Okay, so that may be, and this is what these Chinese researchers did. They analyzed both the layers because, like I said, they put down the lower layer first, the larvae sit on it, and then they eat the top layer, and then after they're capped over, then they eat the bottom layer. So that's a possibility. It could be that the specific ratio may have been off, and maybe they didn't get the right amount of sugar at the right amount of time.
Okay.
Last question here. Oh, go ahead, Jason.
I was just going to say, I'll go back and try that again next spring. I have one more after you do this last one, though, Joe. I thought of something else when he was talking about that.
Well, go ahead right now, and I'll get the last one.
Okay. Queen mandibles.
Yeah.
Multiple queens in a colony.
Yeah, by ablation of the mandible.
Exactly. Have you seen that? You never heard of? Okay. Have you tried it?
No, I haven't.
Okay. Well,
but it certainly sounds interesting. You cut one mandible off a queen, and somehow they're able to realize that they're damaged, and they tend not to fight. So that's pretty. That kind of perception is pretty amazing. Nobody quite figured out how that works, but the Chinese use that for their royal jelly producing colonies.
Right. I tried that with a handful of colonies. I introduced double queens, clipped a mandible on both of the queens in each colony, and my thought was for brood production, but they always seem to pick one and get rid of the other one.
Oh, okay.
But I didn't have very good luck at it. But again, I didn't have the full story. A lot of the stuff that you see done in China and other places, unless you can read Chinese, you can't get a whole lot of information on it.
Yeah, there's also a single study is just that. It's a single study. Science is all about replication. So until you see something, and this is the problem, we go back again, that question of the stuff that gets repeated over and over again, that's maybe based on a single study, and you think, you just keep repeating it without anybody ever duplicating the study. So I question a lot of that stuff that's based upon a single study.
All right. Have you seen the queen jackets that they're working on now for overwintering multiple queens in a colony, loose in a colony?
No, I haven't. Okay.
There is, I know an entomologist in Ohio, and she is Chinese, and she has some friends back in China that are working on this, and I've seen pictures of it, and it's some kind of a little flexible jacket they just put over multiple
geez.
queens, and
Yeah.
they'll have like 20 overwintering in a colony.
Oh, wow. There you go.
But yeah, that's something I asked her to get me more information on, but I haven't. Then
Well,
COVID came.
ask her, like,
gotten...
when they're dressing them with those jackets, do the queens cooperate and put all 6 legs into the jacket sleeves?
Yeah, I imagine not. But I'm going to get more information from her.
Okay. Last question here. Dan Leitner, unmute yourself and you're up. Dan, you there? Evidently not. Randy, it's been a very much of a pleasure. I've learned a lot, answered some questions that I couldn't get answered. I'm sure that other people's had questions that they never could get answered, and I really appreciate the time.
All right. Well, good luck with the beekeeping, guys. We're getting a warm day here, and there's pollen coming in today for the first day. So very exciting in our build up for taking our bees to almonds to see some pollen coming in 4 weeks before the bloom. That'll help us a lot.
Well, thanks for everybody for coming, and we'll try to do another one. Good night, guys.
All right. Take care.
Thanks, Joe, Randy.
All right. See ya. You bet.